Hello students, in this video we will be learning what are the features of a cloning vector or a plasmid. One example of a cloning vector called as PBR322 and its structure, which is a possible question for your CBSE board exam. Alright, let us look at what are the features of a plasmid. There are 3 points that you will write under this question, features of a cloning vector. First one is that it should have a origin of replication. Why is it that required? As you know, these are the plasmids, let us say, this should be able to make its own copies by itself without the help of genetic material, right? So for this, there has to be a sequence called as OD or origin of replication. Otherwise, this DNA cannot make its own copy. In other words, it cannot replicate. So in order for replication, origin of replication is required. In other words, ORI is required. So first point under this is, under the features of plasmid is origin of replication should be present. Point number 1. Second is site for common restriction enzymes like EcoR1, HIND2, BAMH1, etc. should be present on the cloning vector, as well as it should not have multiple recognition sequences. What do I mean by that? Let us say EcoR1's site is here on this plasmid, which is given as an example in our textbook. EcoR1's site. What do I mean by site? If I add EcoR1, in other words, the EcoR1 restriction enzyme to this mixture, to a mixture having a vector like so, it will go and cut over here. So its recognition sequence is this one. All right. Now let us say instead of here, there is one more recognition sequence for EcoR1. Let us say it is here, right? There is one more here. Now, the moment I add EcoR1 restriction enzyme into this, what will happen? It will go and cut here also, resulting in multiple fragments. We don't want that. We just need a single place where my enzyme can cut. Clear? So that is what is meant by have a single recognition site for the restriction enzymes, common restriction enzymes like so and so. Third point is that it should have a selectable marker. We have learned in the previous video what is meant by a selectable marker. It is an indicator gene present on the plasmid which shows whether the host cell is a transformant or not. Students, as you are also expected to know the structure of this cloning vector called as PBR322, I have labeled 4 things on this vector. Let us see what all are the things that you need to label. This labeling can look very confusing. Let's simplify it in our own way. That is, first and foremost, you're going to draw 2 concentric circles like so. Label 4 genes. That is it. First one is called ORI. Here, leave some space blank. ROP, second gene. Blank, tetracycline resistant gene. Blank, ampicillin resistance gene. That's it. So basically, you're just drawing 2 circles. Number 1 gene, blank. Second gene, blank. Third gene, blank. And fourth gene, blank. That's it. Done. Number 1 is ORI. 2 is ROP. 3 is tetracycline resistance gene. 4 is ampicillin resistance gene. Once you're done with that, we have to remember which gene is having which recognition sequence for which of the RE or restriction endonucleases. Okay. So here, if you look at ORI, nothing is there. No, it is not having any restriction enzyme site. It is not going to be cut with any of the restriction enzymes. ROP can be cut by PVU2. Tetracycline can be cut here or here depending on which restriction enzyme we are going to use, SAL1 or BAMH1. The ampicillin resistance gene can be cut by these two. The space between AMP and TET can be cut by any of these three, EcoR1, CLAV1 or HIND3. This you need to put in little effort and learn. For that I have made a table for you. You can take a screenshot of this or write it down in your notebook and learn it up. So this is your table. That's it. Inside ORI, nothing. ROP, PBU2, tetracycline resistance gene, BAMH1 and SAL1. Ampicillin resistance gene, two of them, PSP1, PVU1. Between the sequence of AMP and tetracycline resistance gene, three of them, EcoR1, CLAV1 and HIND3. This table, learn it, that's it. That's your 2 marks in your board exam. It's a probable question, so please put in little effort and learn.